The second part of Chapter 6 deals with different types of media. Some of these we'll be working with later this week because they're really helpful in identifying or differentiating between different types of microbes. And so our learning objectives for this section are to differentiate between what we call chemically defined and complex media, looking at some of the different types of media and their uses, and then finally, what are some methods of handling and preserving microbes? So when we talk about a culture medium, these are nutrients prepared for microbial growth. We have used a lot of what we call nutrient agar. That's very common, general purpose agar that will allow the growth of most microbes. And we always try to make sure it's sterile before we start out. That means that there's no living microbes on it. That's why I always autoclave. I cook up the agar. Basically, you make it up almost like Jell-O. As you have this powder, I mix it in distilled water. We cook it in the microwave until we bring it to a boil, so that it basically dissolves in the water. But then I put it in the autoclave and bring it up to a particular steam and pressure for 15 minutes, and then I pour the plates. Or if we already have them in the tubes, we just pull the tubes out so they can cool. Because we want your media to be sterile with no microbes, so that when you put your microbe on it, it should be a pure culture. When we introduce microbes to a medium, we call that the inoculum. That's why I always say you're going to inoculate your broth tubes. You're going to inoculate your phenol red tubes, those types of things, because the inoculum is the introduction of that microbe to the medium. And then the culture is when the microbes are growing on the culture medium. So these are just some terms you'll need to know as we go through. Our goal is always to have a pure culture. We don't want a contaminated culture where we have unwanted microbes in there. And we also don't want a mixed culture. Even if we know what the two microbes are, we would like to have an isolated culture. So a pure culture contains only one species or strain. So a colony is a population of cells that arise from a single cell. And sometimes we call a colony forming unit. And the streak plate method, which you have used, is used to isolate pure cultures. So by streaking it here, and then pulling a little bit here, and then pulling a little over here, hopefully you're going to end up with these nice isolated colonies by the time you're done. Doesn't always work if your microbe is a very prolific grower. Sometimes all you'll get is big smears all the way across because they're just they just grow so heavily, you can't get that. So again, we often will use agar. It's a complex polysaccharide. It's used as a solidifying agent. We use it in petri dishes, slants, and what we call deeps. It's nice because it liquefies at 100 degrees Celsius. Okay, so I can bring it up to boiling and get it liquid. I can autoclave it at that high temperature and steam. But then once I pour it into the petri dishes, it solidifies at around 40 degrees Celsius. And so we can grow our microbes on there and not have to worry about it melting when we put it in the incubator or anything like that, because we're always keeping our incubators at 37 degrees Celsius or less. And it's not metabolized by microbes, so that makes it very useful. Microbes aren't going to break it down and eat it. They might get food from it, but they're not going to destroy the agar, and it's going to stay solidified for us. So we can break culture media down into two basic groups. Either chemically defined media, that means we know exactly the chemical composition. These are generally used in research labs. Where you have to know the exact chemical composition of what's going into that. The other type of media is what we call complex media. A lot of times you'll see the word extract in there, where there'll be extracts or digests of yeast, meat, plants, let's say beef extract, beef peptones, or whatever. Nutrient broth, nutrient agar, these are both complex media. They're just basic. We can grow most microbes in and on them, but they are complex because they have extracts in them. So if you look at something like this, here's a chemically defined medium. We know exactly all the stuff here, every little category here of what's in it and how much is in it. If you look at the next one, for a complex medium, look at this. Peptone, it's a partially digested protein. Beef extract, okay? It's giving you the amounts, but we don't know, well, what's in beef extract? What do you mean by that? So it's more complex. It's not chemically defined. We don't know exactly the composition of it. Some microbes, as you found out, are anaerobes, either obligate anaerobes or maybe they're facultative anaerobes, and so they might grow best if you use a reducing media. This contains chemicals that combine the oxygen, and then we heat it up and it drives off the O2, so it creates a anaerobic environment. Or at least a reduced amount of oxygen anyway. So we like to see, based on where the microbe is growing, is it that obligate anaerobe, is it an obligate aerobe? And so we're going to use thioglycolate tubes, and you'll be inoculating those later this week to see how your microbe grows and if it likes oxygen or not. We can also create an anaerobic condition using anaerobic jars, or in some places, in some of the either hospitals or research labs, they actually have these anaerobic chambers here where you just kind of use gloves and reach your hands in and kind of work with the microbes that way. Capnophiles require high CO2 conditions, so those are microbes that actually need to have higher carbon dioxide conditions. And getting into various different types of media now. Selective media suppresses unwanted microbes and encourages the growth of desired microbes. An example here is mannitol salt agar, which we abbreviate MSA. You'll be using that this week. It has a very high salt content in the agar, and that suppresses the growth of most microbes. So when you guys use this, a lot of students say, oh, I did mine wrong. I didn't have anything grow on my petri dish. Well, it's not that you did anything wrong. It's just that your bacterium can't handle high salt conditions or that really high osmotic pressure. So it just couldn't grow on it. It doesn't mean that you goofed up or anything. Mannitol salt is also what we call a differential media. Differential media distinguishes between different groups of microbes. In other words, if microbes can grow on it, they're going to look different. So it differentiates between microbes that ferment the sugar mannitol. So fermenters turn pink-red agar to yellow, and then the color doesn't change for non-fermenters. So nothing was on this right here, but Staphylococcus epidermidis, it could grow on the salt. That means it can grow on that selective media. It can withstand that high salt concentration. But Staph aureus, a different Staphylococcus, can ferment the mannitol, so it turns yellow color here. So it's kind of differential as well. Differential media allows different microbes to grow, but usually includes color changes to distinguish between different types of bacteria. So instead of suppressing some and encouraging others, it might allow different things to grow, but those different things may look different on there, and that's what helps you differentiate between them. So a very common one we use that's differential is eosine methylene blue, or EMB. So it might slightly inhibit the growth of gram-positives, so you'll have to see if any of you that have gram-positives, if yours grows on it or not. But if they are able to, gram-negative are able to grow on it. They may appear kind of dark purple. Some of them, such as E. coli, create this really pretty metallic green sheen, and other fermenters produce larger colonies that are only purple in the center. So if they're able to ferment the lactose to produce an acid end product, they might have some of these different colors in the end, and that's what differentiates them. That's why it's called a differential media. So there you can see how pretty E. coli looks with that metallic green sheen. Others grow on it just that kind of dark purplish color. Enriched media contains nutrients that encourage the growth of desired microbes. These are often used for soil or fecal samples. Chocolate agar, yeah, it sounds good, right? I love chocolate. Not this, because it's cooked blood agar. It just kind of has that brownish color to it, kind of like chocolate. So not very appeasing there. But some bacteria, which we call the fastidious bacteria, or the picky eaters, really like that because it has some of these special nutrients in it that really encourage it to grow. So sometimes if we're having trouble growing microbes, they might use a special enriched media such as this cooked blood agar to see if they can get the microbes to grow. Are any of you fastidious? Are you picky eaters? That's going to be your new word for the day, fastidious. Other types of media that you're going to be using this week, Simmons citrate. It tests whether a microbe metabolizes citrus or not for an energy source. So this is what it normally looks like when I pour it. It's kind of a greenish color, sometimes a greenish aqua color. So if it does not metabolize it, it's just going to stay the same color. So that's a negative test result. But if it turns this nice, almost cobalt blue, that's a positive test. That means it does break down the citrate for its energy source. The starch plates are very cool. Basically, you will grow your microbe on there, and then the next day, once you see growth, you put a whole bunch of iodine on this plate. Well, if you remember from biology, iodine is an indicator for starch, and it will turn this kind of dark purplish black, all right? Well, if your microbe can break down starch or hydrolyze it, then there won't be any starch left in that agar, and then when you try staining it with iodine, it will make this like halo, these little bare spaces around your microbe. That's a positive test for that. The methyl red Vogts-Proskauer test, that's a new one we're going to try this year. I haven't tried it before in class, but it's used to determine which fermentation pathway is used to utilize the glucose. So there's two parts to it. You do the methyl red test. If mixed acid fermentation pathway is used, the glucose gets fermented and it produces several different acids. A red color will indicate a positive result. Basically, you'll grow your bacteria, you'll inoculate it in the culture, so it grows in this broth. The next day, you add some methyl red to it. And if it does stay red, that means it's a positive result. If a yellow color is there, it indicates a negative result. Okay, don't get this confused with our phenol red tests, because they're just the opposite. When we use phenol red indicator, if it turns yellow, that's a positive test for fermentation of those particular sugars. And if it stays red, it's negative. So this one's exactly the opposite, the methyl red test. So try not to get that confused. Next step is the Vogts-Proskauer test. So that's to see if they use this 2,3-butanediol fermentation pathway. I don't know how to pronounce that. Okay. So they're looking to see if they produce this instead of the acid end products. So now we use a couple of other chemicals, naphthol and potassium hydroxide, and they're mixed together. And so then what you're looking for is the presence of acetone, which is a precursor of this. And so if the culture is positive for the acetone, it'll be kind of a brownish pink color. But if it's negative, it'll turn kind of more of a coppery, brownish green to yellowish color. So again, a positive test if it's more red versus the more yellowish, and then positive here versus the more brownish, greenish, yellowish here. So when we handle microbes, we need to be careful. We need to look at various biosafety levels. If it's a biosafety level 1, you really don't have to do any special precaution. If it's a biosafety level 2, lab coat, gloves, eye protection. So when we think about in lab, this is pretty much where we're at with all of our microbes. You still have to use good aseptic technique. You should always wear your goggles, put gloves on. We don't wear lab coats, but you could put on, kind of your lab apron if you needed to. If it was biosafety level 3, then you'd probably work in those special hooded kind of cabinets. You might have to wear specialized masks and suits. And so a lot of the time, I think with COVID, and I'll have to double check on this, I think it's at a biosafety level 3 that a lot of times you saw on TV, at least when they were really in the throes of things, a lot of times they were wearing these biosafety suits just to make sure that people were getting infected by it. But if it's biosafety level 4, then you're sealed in a negative pressure room with specialized suits that hook up to their own air source. Negative pressure means the air is being drawn into the room rather than filtered out, because they don't want this microbe to escape into the outside world or into another room. Things like Ebola virus, things that are very lethal, that could spread easily, that could cause a lot of problems, those are more of the biosafety level 4. And see how they have to work with special suits with their own oxygen supply there hooked up to them. In terms of preserving bacterial cultures, they can deep freeze them, or they can do what we call lyophilization. Lyophilization is freeze drying. So they basically dehydrate the bacterium, and then it's frozen. Sometimes when I've ordered microbes, I've gotten them lyophilized, and then all I do is, add some, like maybe some liquid nutrient broth to it, and boom. Once I rehydrate them in that liquid broth, then they start growing again. So that's kind of a nice way to store and to ship bacteria, especially in our area where you don't want glass vials and things to get frozen and to break. Shipping them lyophilized is a good way.